Technology 100 slide. This is an interesting question. When should you guys learn about early life and how life started? You didn't learn about that in 211? Did you learn about it in 211? Maybe you already learned. So, why are plants photosynthetic and animals are not? Anybody know?
Cyanobacteria.
What's that?
Cyanobacteria evolved to plants.
Oh, very good. So the first photosynthetic organisms were bacteria, right?
Yeah.
Cyanobacteria. To this day, cyanobacteria still do a lot of photosynthesis. About 2/3 of the photosynthesis on the planet is not done by plants, it's done by protists in the ocean. So what happened was, if we go back, way back, to the origin of single-celled life, origin of prokaryotes, there was a split in the lineage. One lineage went on to become what we call the true bacteria, things like E. coli, cyanobacteria like that. Right? The other one branched off and some became the eukaryotes, and some became the archaea. You guys learn about archaea? Archaea like methanogens. Well, what happened was part of the bacterial lineage, some member of that, was engulfed by eukaryotes early on, and that became the mitochondria. Okay? That lineage went on for a while, and then there was a secondary endosymbiotic event in which chloroplasts were taken up again, okay? And that gave rise to the plants and the photosynthetic protists. And notice there is a polyphyletic grouping going on over here. Nobody's quite exactly sure how this works. The protists, if you could get research grants to study protists, you would always have money because nobody understands these things. They're just a very strange grab bag of stuff. Anyway, this lineage here that's of the eukaryotes with just the mitochondria went on to form the fungi and the animals. So fungi are actually more closely related to animals than they are to plants. Okay. Anybody know what the fundamental difference between a fungi, a fungus, and an animal is? It's the way it eats. So whereas you and I eat by putting food into ourselves, fungi eat by growing into their food, right? Fungi digest externally. That's how they eat. Okay. Cool. But anyway, then here are the plants down here. The plants, get the mitochondria and the bacteria. Sorry, mitochondria and the chloroplasts. Sound cool? So in the same way that mitochondria has its own DNA, so does the chloroplast. Chloroplasts have their own DNA as well. Coolio? All right. On to dermal tissue, the dermis, the skin of the plant. Okay. So, the epidermis is the outer covering of the plant. I pointed this out before. It's this layer of cells on the outside. So the epidermis is a tissue because it's made up of epidermal cells. It excretes the cuticle. Cuticle is a waxy layer that seals the plant and keeps water in, so the plant doesn't lose water. Part of the dermal tissue are also the stoma or the stomata, works either way. And these are the holes in the leaf, okay? The structure of the stomata is made up of two cells called guard cells. guard cells because they guard the opening. That is the stomata. And this is where gas exchange occurs in the plant. What gas goes into the plant? Carbon dioxide. What is one gas that comes out of the stomata? What's the other one? Water. Water. Good. Okay. So other specializations in the dermal tissue, root hairs. So roots are absorptive structures. They absorb water and nutrients out of the soil. The greater the surface area, the more they can absorb. How do roots increase their surface area? They grow these really tiny, tiny little hairs that are able to take in even more water. Plants have gone one step beyond that by having a symbiotic relationship with fungi that actually, the fungi infects the root hairs, and the hyphae of the fungi are even smaller than the root hairs themselves. And they create an even smaller, larger surface area to absorb more. That relationship is called mycorrhizae. We'll learn more about that later on when we talk about plant nutrition. Okay? So here's some cross sections. So here's the epidermis, there's the cuticle, this is the stomata, this is the guard cell. And you can see how the cuticle has sort of covered in the area and filled in the gaps around the guard cell. That's cool. Here are the root hairs, so this is the root itself, and these little hairs coming off like the hairs you have on your arm, right? And that's where water and nutrients are entering the plant. Kind of cool. And small, 160 microns, right? So 1,000 microns is 1 millimeter. So these are small little tiny structures. If you ever transplant a plant, like you move it from one area to another, it shocks, it's damaging the plant largely because it's destroying all these root hairs, and the plant has to regrow those. Which is why when you transplant, you want to give the plant a lot of water. So it makes it easier for it to absorb because it's lost so many of those root hairs. Okay, some of the stem structure. A lot of this is just like terminology that's good to know when we talk about a plant and that sort of thing. So plants grow, we think of plants as growing from the ground up. They actually grow from the top down. So the growth of a plant, you know, in humans we have those growth plates that we grow from. What are they called? They have a term for them. Growth plates, yeah, but it's just growth plates. There's those axes, right, that your bone grows off. You know what I'm talking about? Those growth plates. They're growth plates. Okay. In plants, the growth plate of the plant is what's called the terminal bud, okay, or the terminal meristem. We'll learn about this in a minute here. The meristem is the area of cell division in the plant. And as those cells divide, it causes the plant to get bigger. So at this terminal bud, where this apical meristem is located, as the cells divide there, that's what's creating all of the stuff underneath it. In doing so, that pushes the plant upward. Does that make sense? So it's actually, the growth is coming from that location up there, from that meristem. All right? All plants have these apical meristems. They have one in the top, here at the terminal bud at the top of the shoot, and they have one in the root as well, that causes the roots to go down. Some plants have what are called lateral meristems. Lateral meristems, we'll get into those later on, they create wood, they cause the plant to grow outward and to give it girth. Okay? All right. We have branches here. Branches, you know, this is where the leaf branches off at what's called the node. Don't worry about that so much. Internode is the space between those two. Probably all we need to cover there. Leaves here. That's a tomato. Okay. All right. Vascular bundles. I mentioned vascular bundles before. talk about transpiration and translocation. Give me a second there. Which are the processes of moving stuff through plants. We'll see that the xylem and phloem are always next to each other. And the reason for that is the phloem needs the water from the xylem in order to work. So the xylem and phloem combine together to form these bundles, these vascular bundles, these bundles of vascular tissue. Okay? In monocots, remember, in dicots, remember, these vascular bundles form a ring around the central pith, the ground tissue of the plant. So for example, right here. So these vascular bundles. In the middle, so this is one vascular bundle. This is the xylem here that's causing the water to move. The water is moving through there. And next to it is the phloem, and that's where the sap is going to move, inside that vascular bundle. Okay?
So we surround the ground tissue.
Yep. This part here is the ground tissue. Yeah, it makes up, it's structural, makes up the structure of the plant. Okay? Called the pith. The area outside of the vascular bundles is called the cortex. Remember that term cortex. We're going to run into that again when we talk about trees and bark. The cortex becomes important because it's what produces the bark. Don't worry about this steely thing here. Don't worry about that. In monocots, remember that the vascular bundles are spread throughout the stem. They're not found in a ring. They're just scattered throughout. All right? But you still have in the vascular bundle, you still have the phloem on one side and the xylem on the other. Phloem and xylem, again, they need to be next to each other because the phloem needs the xylem in order to function properly. You can actually have xylem on its own, as a matter of fact. This right here is pure xylem. There's no phloem in there. That's just pure xylem. Wood is secondary, called secondary xylem. It's just all xylem. Which means, remember xylem is dead at maturity, means wood is dead at maturity. So in a living tree, the actual wood inside of the tree is totally dead. Cool thing about a tree, the bark of a tree is dead, the wood is dead. The only living part in the trunk of a tree is this little layer in the middle here, which includes the phloem and the cortex and what we call the vascular cambium right in there. The rest of it's dead, which is why you should never carve your initials in a tree. Because if you break through that outer barrier and break through that living part of the tree, you allow pathogens into the inner wood of the tree, and there's nothing in there to stop them. Right? So the pathogen can just spread up and down the tree and kill it. Okay. All right. Cool side note, this is one of these things, don't really know where to throw it in. I'm going to throw it in here. Talking about stems, some cool adaptations to stems, it's kind of neat. Tubers are actually stems that grow underground. Onions are stems, they're actually leaves, right? Tubers are stems that grow underground. Tuber, potatoes, sweet potatoes, yams, tubers. Strawberry stolons, runners, strawberries, they run along the ground, they can spread like that. Those are stems. They look like roots, but they're actually stems that grow like that. Okay. Leaves, we talked a little bit about in lab. The petiole is the thing that supports the leaf from the stem. Okay, the node is where it's attached, mentioned that already. The main part of the leaf is called the blade. In monocots, because you have the leaves grow like this, they've got those parallel veins on them, there's a sheath that clasps onto the stem. One of the ways to distinguish it. I'm just introducing this stuff, don't worry about it too much. This is mostly going to be used in identification of plants when we go on our field trip next week. These are all what we call simple leaves. They only have one leaflet. This is a spade-shaped leaf, like if you play cards, spades, right? The margin of the leaf is toothed. You see it's got these little teeth on it? These are lobed leaves because there are these sort of big bulges to the leaf, right? This is an oak tree, and this is a maple. These are lobes as well. What's the difference between the two of them? Couple things. One, notice this has one central vein with branches coming off of it, whereas the maple has the veins branch out from this central point here. You see that? You see the difference there? Okay. The other difference is the edge of the leaf. The maple has a toothed edge, whereas the oak has a smooth edge to it. Again, these are all things that we use to help identify. These are compound leaves. So this is one leaf with 1, 2, 3, 4, 5 leaflets. This is what's called palmate leaf compound, so it's like a hand. This is black locust with this leaflets coming off. This is what's called doubly compound. So this is one leaf with all these little leaflets coming off. Well, when we identify plants, we'll get into that more. All right, now we actually look inside of the leaf. We can see where the gas exchange occurs. So leaf structure is, it's pretty interesting. It's probably the most dynamic structure in a plant. I mean, truth be told, I love plants, but they're not nearly as dynamic as animals. Animals are just way more dynamic. They move. They eat things. We eat them. What's that? Plants eat things? Some plants do. Okay, so inside of the leaf, we've got two major parts. Well, we have what's called, we have the mesophyll, which means middle of the leaf, right? Meso, middle, fill, okay? We have the spongy mesophyll because it looks spongy. Right? Here, spongy. And then we have what's called the palisade mesophyll. The palisade mesophyll is where the majority of the photosynthesis is occurring. Therefore, the palisade mesophyll tends to be on the top of the leaf because that's the area that's getting the sunlight. These are what are called palisade parenchyma cells. They're packed together closely. They do lots of photosynthesis. They're in charge of photosynthesis. The spongy mesophyll, on the other hand, is for gas exchange. Notice the amount of surface area. Notice the space between it. Okay? This is allowing the CO2 to come in here and interact with the cells. It's allowing the water to leave out of the stomata and allowing oxygen to leave as well. The water is leaving because it's being brought up here via the xylem, okay? And it's being taken to the cells for photosynthesis, and it's actually just going straight out the stomata. We'll learn about this when we do transpiration. But what's happening is the water is being pulled up the leaf through the stomata. That process of pulling it carries the water along for it to be used everywhere else. Next to the xylem is the phloem, right? So these are the tracheids, these are the sieve tubes. The sugar being made here in the leaf is being pumped into these sieve tubes and taken down someplace else. It could be going to another branch, it could be going to the roots, it could be going someplace else. All right? Questions on that? That was kind of a lot to digest all at once. Any questions? No? This is an electron micrograph, what that looks like. Pretty good. Pretty good description. Okay. Roots. Let's skip that. Roots also have an apical meristem. This, we left it like near a heater and it just like melted. So, roots also have an apical meristem, so the root grows from this point, right? And it separates out and it causes the root to grow down deeper in the soil. Now what's neat about roots though is whereas the shoot apical meristem is just growing up into the air, roots have to grow through the dirt. So what they do is they produce a bunch of cells that are very loosely attached to the plant, and it creates what's called a root cap. And what that does is that lubricates the root as it passes through the soil. Those cells get sloughed off, and it helps the root to move through the soil. Kind of cool, because it has to push its way through. And roots are very, very strong, right? Roots will actually crack open rocks. Roots will push their way through pavement and the sidewalk. You can see that stuff, right? Roots are phenomenally strong. Okay, so here at the apical meristem, that again is where the cells are dividing, and they're producing the basis of all of those primary tissues. They're producing the xylem, the phloem, the epidermis, the cortex, and all that kind of stuff, which is eventually going to grow up into the rest of the plant. What's neat, I think, is that you can actually draw a straight line from the xylem that are created here in the root all the way up to the xylem that comes out the leaf. It's the same exact tissue, the entire length of the plant. Okay, we're talking about growth. Let's talk about how plants grow. So you and I experience what's called determinate growth. Determinate growth means that you grow to a certain size and you stop. Most animals grow this way, though not all. Some animals keep growing and growing and growing and growing. Halibut do that. You guys like to eat halibut? Right? The older they get, the bigger they get. Sturgeon are like that. The older they get, the bigger they get. Great white sharks are like that. They just keep growing and growing and growing. Okay? Plants, on the other hand, have what we call indeterminate growth. They grow until they die, right? So the biggest tree is always the oldest tree. It just keeps growing and growing and growing and growing and growing. All right, does that make sense? Now some plants don't live, plants don't live forever, so they eventually stop growing. Some plants don't, live very long, so they're down.
What about the oldest tree, Methuselah?
What's that?
What about the oldest tree, Methuselah?
It's just growing very slowly. We know it's the oldest tree because we met, you heard the story about the dude who discovered this? This poor schmuck. He worked for the Park Service, right? So they were looking at these bristlecone pines in the Great Basin somewhere. I don't know, it was Arizona, California, things like that. And they're doing let me tell you about something. You can age a tree by its growth rates, right? So they want to know how old this tree was, right? So he cuts down the tree and he measures the growth rings and discovers it's the oldest thing ever. Like he had killed the oldest known living organism on the planet.
Why didn't he want a course?
So you cut the whole thing
Way to
down?
determine the
Yeah, they didn't have one of the problems is the core samples only work on relatively soft wood. And because these things grow so slowly, this wood is phenomenally dense. So you can't actually do a core sample, right? And there were a bunch of them, right? They're like up there all over the place. It was like, I'll cut one down and then you realize like, oh shit.
It's gone now, it's dead.
He killed it, yeah. Now there might be older ones.
We're not gonna know that by now.
Yeah, right, yeah. That sucked.
Different way of doing a course.
Oh,
Might they find a different way of doing a course sample?
this is back in the 70s. They may have a different technique now, but yeah. I mean, they might, I don't know. And maybe, yeah. Okay, so plants grow from their meristems. Meristems are just, it's regions of cell division, right? So cells are dividing and dividing and dividing, and that's what's causing the plant to grow. So for example, this is kind of illustrated pretty well here at this root apical meristem. You can see that the cells that are closer to the meristem are smaller because they're dividing and dividing. But then as the cells mature, the cells also get bigger. So plants grow by an increase in the number of cells, but also an increase in the size of the cells. Both of those cause the plant to get bigger. Does that make sense?
Wouldn't that reduce density?
Yes.
Don't older trees get denser?
Not necessarily. I mean, do older trees get denser? No, not necessarily. Like, it depends on the tree. We'll look at this in a second. Those bristlecones grow really, really slowly, and so they tend to be very dense. But like Doug firs and a lot of the conifers, as they get older, they get kind of soft because they're big. Okay. So the primary growth of the plant comes from the apical meristem, which I mentioned before, at the shoot and the root, okay? And so what's going on there is there's division occurring, and that's causing the plant to grow down from the shoot and then up from the root, and that causes the plant to get bigger and bigger and bigger. All right? And it basically just causes the plant to grow up and down. Out somewhat, but mostly just up and down. Okay? Like that. Some species only have primary growth. Our weedy species, our herbaceous species, things like dandelions, they only grow primarily. That's it. Okay? They don't grow bigger. Trees, as we'll see in a second, actually get bigger. They have what are called lateral meristems, which cause them to go. So, primary growth comes from the apical meristems. Other cool thing about plant cells. Living plant cells are what are called totipotent cells. What's that? Yeah, totipotent. They're like stem cells, right? So all plant cells are basically like stem cells, meaning that they're undifferentiated and they can be differentiated into a different cell at a later point in time. This is why you can take like a branch off of a tree, stick it in the ground, and it'll actually form roots. Those branch cells can become root cells. Plants have that plasticity. Most animals, most of the time, can't do that, right? Our cells become differentiated relatively early on in our development, and then they're always that, right? Your liver cells will always be liver cells. Your skin cells will always be skin cells. You have a few stem cells as an adult, but not very many. What's up, Peter?
Well, I saw this on a video where people were actually trying to reverse that. So
Mhm.
take skin cells and make them back into stem cells.
Right. Yep. We're trying to flip that. But like, we have to do that. They're not naturally capable of doing that on their own. Yeah. Yeah. We do actually have some stem cells in our fatty tissue has some stem cells there. Okay. All right. So there are 3 primary meristems. For each of the tissue types. Okay. There's the protoderm, which produces the dermal tissue. There's the procambium, which produces the xylem and phloem. And then there's the ground meristem, which produces the ground tissue. And they all come from that apical meristem. You can remember protoderm, proto before derm, dermal tissue, procambium. The cambium, as we'll see here, gives rise to the vascular tissue. And then the ground meristem. It's the meristem for the ground tissue. It'd be nice if they could like have like a consistent naming scheme. Okay, so here's how this works. All right. So here is the shoot apical meristem. So all the growth is coming from that region there. Okay. Those cells divide, and as they divide, they differentiate into the procambium, right here, which will eventually divide into the xylem and the phloem. What's going on, this is kind of cool. So this procambium here, it's dividing itself. If it divides on the inside, that becomes xylem. If it divides on the outside, it becomes phloem. So as a plant keeps growing, it's making more xylem and phloem from the procambium. Okay? Is that all I want to say about that?
What?
I think that's all I want to say about that. What's
What's
that?
Becomes the ring.
What becomes the rings? We'll get to that in a bit. That's actually caused by the lateral meristem. This is what's called secondary growth. So primary growth of the plant is up and down. Secondary growth is what we call girth. It's not so much side to side as it is getting fatter, like me at Christmas, okay? Again, not all plants do this. These lateral meristems cause the plant to grow outward. They cause the diameter to get bigger. Okay. So you have these lateral meristems here that are causing the plant to grow out, causing it to grow away from the lateral meristem in both directions. This is how wood is formed. So wood is secondary xylem created by the lateral meristem. And what happens is, we'll get to why there are rings in a second, but basically it's growing out like this. Now notice, the stuff that grows outward gets to keep growing out and out and out. The stuff that grows inward gets compressed more and more and more. And so if you look at the center of a tree, kind of shows it here, you can see it here even better. See how it's really dark? What's happening is that the metabolites in the wood and ions and other things that are being carried in the xylem get stuck in there. And they end up filling that all up. It gets kind of full of gunk. And that's why the center part of the tree has that dark color to it. It actually gets so stuck that it won't carry water anymore. It becomes what we call the heartwood of the tree, right? But basically what's happening is it's being compressed and compressed by the growth of the lateral meristem. It's just pushing inside and inside and inside. The stuff on the outside can keep growing because it's just growing outward. Now, as it grows outward, the bark that's the protective coating on the outside has to keep up with it, but it can't quite keep up with it, so what happens to the bark? It splits, right? And so that's why we see those splits, and different barks split in different ways, and you can oftentimes identify a tree based upon the pattern of the splitting of the bark. Okay, so secondary growth occurs at the vascular cambium, which produces secondary xylem and phloem. Secondary xylem is what we call wood. Yeah, what's up?
Is it kind of the same in a carrot?
Yep. Sorry, that's primary growth, that's not secondary growth. Yeah, that's just primary growth. Same basic thing happens, yeah. Yeah, the stuff on the inside gets compressed. Yeah. Okay. So So the vascular cambium produces that secondary xylem and phloem. The cork cambium produces the cork, which is part of the bark. So the vascular cambium is one of the lateral meristems. It makes that secondary xylem, which you and I call wood, and it makes secondary, my handwriting suffering, phloem. Okay. Cork cambium. This makes the cork, which leads to the bark. Okay, I'm not gonna go into that. So in a woody tree, you have the vascular cambium here, right, which is a secondary, which is a lateral meristem, producing secondary growth. It grows inward and outward. On the inside, it always produces xylem. On the outside, it produces phloem, right? That xylem builds up and becomes the wood. This, the secondary phloem, is it's underneath the bark. It's actually, like I said, it's the only living part of the trunk of a tree. And then here's the cork cambium, which is producing the cork and then the bark on the outside of that. They're both growing simultaneously outward. And they cause the tree to grow in girth. I don't have enough of these for everybody, but take a piece of wood and pass it around. Just take it. Pass it. Let me keep on this. Okay, so if we think about what's happening here, very good to see one. Okay, so you can think of the cambium as it's a tissue, but it's really just a single layer of a cell, right? One cell. What it's doing is it's dividing. That one cell divides into two different cells. The cambium cell, the one that, that will always remain a cambium cell. But the cell, either on the inside or the outside, that differentiates into xylem if it's on the inside, or it becomes phloem if it's on the outside. Okay? So this becomes xylem, this cambium splits again, divides again. This one on the outside now becomes phloem. Okay? That cambium is going to divide again, but notice that first xylem is now on the inside. Okay? And the newer xylem is closer towards the cambium. So the older wood is deeper into the tree, right? And the newer wood is farther towards the cambium. The phloem works in the opposite direction. The older cambium is, sorry, the older phloem is on the outside, and the newer phloem is towards the cambium, right? And that's just because the way it grows, right? Okay. When we measure the age of a tree, we measure from the cambium inward to the xylem. Okay? All right. Now, can you all see your wood? Okay. This is a type of vascular cambium showing secondary xylem. So here's the secondary xylem here. These are the parts of the vascular cambium. These are what are called rays. Rays move into and out of the wood. They're structural. So these cells are going this way, right? So they're going out like that. This over here is the phloem. Cambium's right there. All right. So vascular cambium producing the secondary xylem on the inside, producing secondary phloem there. Then the core cambium producing the secondary cortex and the bark out there. Okay? As I mentioned before, the heartwood of the tree is that darkened wood on the inside that's getting compressed. It doesn't actually carry anything because it's so full of metabolites and ions and other stuff because it's been so compressed. But it's really pretty and it's very strong. The sapwood, this is what's actually carrying water through the tree. It's the sapwood. Okay. Now, how come we have growth rings? Growth rings only occur in areas of the world that have seasons. Okay? Because what happens? The vascular cambium divides, and then the cells mature and they grow. All right? In the summertime, in the spring, they grow very rapidly, produce lots of cells. They're dividing. In the summertime, they mature, they get bigger, but then, come the fall, they stop growing, right? Because there's no more sunlight and the tree goes dormant. It sits dormant over the course of the winter and then starts growing again. And it goes through that cycle again, making cells that are bigger and smaller over the course of the growing season and then stopping again. Okay? So what you're looking at with the growth rings are lots of cells growing together very tightly, and then they stretch out during the summertime, and then they stop, and the process starts again. In the tropics, where they don't have seasons, it's just hot and sunny year-round, you do not get growth rings. It just grows constantly. Now, this is why people who work with wood like using tropical hardwoods, because the growth rings create areas of weakness in the wood, right? Because they're areas where the growth isn't uniform and they can split more easily. But the tropical hardwoods, things like mahogany, things like teak, things like purple heart, those things just grow all the time. So their cells are super densely compacted together and they're very uniform. Does that make sense? Okay. Now, what's kind of neat is you can age the rings of the tree, right? So you can go back in time and say, you know, like this ring was actually laid down in 1957. This was laid down in 1946, et cetera, et cetera, et cetera. You can go back and count when those rings were produced. I did a cool study when I was in college. I worked with a chemist, and we took a core sample of some of the trees on campus that had been cut down. And we went through and we looked at levels of different chemicals like lead, for example, to see how they changed over time in the tree's history. And you could actually see when automobiles were invented, because the tree was a couple hundred years old, you could see when automobiles were invented, the lead just spiked for a while, and then they took lead out of gasoline and it went back down again. It was pretty cool. Yeah, so you can actually go in and measure the chemical nature of that a tree ring. They do the same thing for analyses looking at like climate change and how global warming has changed over time, because you can look at the nature of the chemical structure of that wood and see what the temperature was on Earth at the time. Yeah.
That's my question.
Yeah. The other way that they do that is by looking at the size of it, right? So if it's shorter, it didn't grow as much, it was probably a colder year. If it was wider, it was warmer. So, yeah. Questions? No? All right. That's it for today. Have a good weekend.